one and all. I would like to welcome all of you for coming on our tour of the wonderful animal cell. On this tour, I'll be talking to you about all the wonderful parts of the animal cells and what their function is. I think the best way to start off our tour is with the cell membrane. As you can see here, the cell membrane is the boundary that the cell forms. It acts as a selective barrier that allows certain materials to pass, but not others. Next on our tour of the animal cell, we'll be visiting the cytoplasm, which is the entire region between the nucleus and the cell membrane. It consists of the cytosol, which is the floating liquid that is the boundaries of the cell. Now, as you look to your right, you can see at various locations of the animal cell that there are large energy sources called the mitochondria. These structures undergo cellular respiration and produce ATP from sugars, fats, and other fuels. On your left, you can see the substance connected to the nucleus, which is the rough ER, which stands for rough endoplasmic reticulum. The rough ER attaches carbohydrates to some proteins and channels the proteins for transportation by the vesicles. The small appendages that you see on the rough ER are known as bounded ribosomes. This is the site of protein synthesis that attaches to the outside surface of the rough endoplasmic reticulum. The bounded ribosomes also produce proteins for use outside the cell as well as inside the cell membrane. And right here, what looks to be free-floating spheres similar to bounded ribosomes are also known as free ribosomes. These ribosomes are also a site of protein synthesis, but are suspended in the cytosol and produce proteins for use inside the cell only. Now, if you look to the side, you'll see a smooth ER, similar to the rough ER, but is different because it lacks attached ribosomes. This structure synthesizes lipids into phospholipids and steroids. It also detoxifies drugs and poisons and stores calcium ions. The structures up ahead are the ventricles, which are paired structures in the animal cell. They consist of microtubules and are presented in a 9+0 arrangement. These structures are involved in cell division. As you look to your right, you'll see the Golgi apparatus consisted of flatted membranous sacs that receive transport vesicles from the endoplasmic reticulum. This structure helps to modify the materials that is delivered from the endoplasmic reticulum. It produces certain molecules as well as lysosomes and secretory vesicles. If you look up ahead, you'll see the liposome, which is a membranous sac of hydrochloric enzymes that help digest food, other molecules, and worn out cells parts. The enzymes can break down proteins, fats, and nucleic acids. These structures function as a recycling system for the cell. They digest the broken organelles. Right here, where you see this large free-floating mass, is a vacuole, which is a membrane sac that stores materials. This structure is larger than the vesicles that you can see on this side. There are two types of vesicles: transport vesicles and secretory vesicles, which Alright. Which the transport vesicles help to carry endoplasmic reticulum products to the Golgi apparatus. The secretory vesicles carry Golgi modified products to the cell membrane and fuses to the cell membrane to dispose of the products outside of the cell. If you look closely right here, you'll see right in between the rough endoplasmatic reticulum and the nucleus is the nuclear envelope. This is a double membrane that forms a boundary between the nuclear contents and the cytoplasm. This envelope is perforated with pores that control what enters and exits the nucleus. Well, ladies and gentlemen, we're nearing the end of our tour. As you can see here, we're at the center of the cell, which is the nucleus that contains most of the genes that control the eukaryotic cell. Generally, this is the most conspicuous organelle in the eukaryotic cell. The nucleus also contains nucleolus and chromatin, DNA, in its center. This is the storage place for all important information that the cell would have. Right here in the center of the nucleus is the nucleolus. This is the place where ribosomes are synthesized and assembled. The ribosomes use the information from the DNA to make proteins for the cell. The liquid that you see in between the nucleolus and the regular nucleus is called the chromatin, which consists of the DNA and proteins, and when it's condensed, it can form chromosomes, which are the building blocks of DNA. The chromatin is important information for cells to reproduce. Well, ladies and gentlemen, we've reached the end of our tour. If any of you were wondering what holds up this animal cell together, it's the cytoskeleton, which is a network of protein filaments that helps the cell to maintain its shape. The cytoskeleton is involved in the many forms of cell movement. There are many types of fibers that make up the cytoskeleton, which as microtubules, such as microfilaments, and intermediate filaments. Thank you for joining us on our tour. I hope you've learned much, and I sure hope that you come again. So long.